Okay, so today I'm going to be talking about understanding DNA. The National Academy of Sciences report on forensic science came out in 2009, and it pointed out a lot of problems with forensic science in areas that are now being discredited, like bite marks and hair analysis, probably fingerprints at some point. But the issues had to do with bias in human examination, with problems with statistics and reporting, as well as the lack of an underlying scientific basis for many of these methods. But they did like DNA and didn't have much to say about it, except that it was a good template for other fields. So let me tell you a little bit about DNA. So the basic biology of DNA is that we're comprised of trillions of cells, most of which have a nucleus. And the nucleus contains the text, the genetic encyclopedia of 3 billion letters written as text in DNA with one complete copy from each parent. And it's subdivided into 23 chromosomes. And so you have 23 pairs of chromosomes, 2 copies of each volume of this encyclopedia. When you open up that encyclopedia and you take a look at one volume and look at a particular page, what you see is text written in a DNA alphabet of 4 letters, A, C, G, and T, and it's read just like linear text. It contains coding regions for genes, and it also contains other regions that have almost no known function and therefore have a lot more variation. And when geneticists and forensic scientists want to understand differences between people in order to track genes or make identities, these regions that have variation are the ones that are used because they're different between different people. The one that was developed in the 1980s is the short tandem repeat. What a short tandem repeat is that somewhere on a page of the genetic encyclopedia, I've indicated on the left what an encyclopedia looks like with 23 volumes. This is half of what each cell has, say, from one parent. A short tandem repeat is a genetic word, usually 4 or 5 letters long, like A, C, G, T, and it's repeated in tandem. So it's a short word repeated in tandem. And what makes them useful is that the number of times that that word is repeated varies in different people throughout the population. So if you have a word repeated 10 times in one individual from one copy they have on, say, the mother's encyclopedia, there may be 15 different lengths in the population. There might be 5, 6, 7, 8, 9, 10, many different lengths. And that's a source of variation that can be used to distinguish people. And scientists measure the number of repeats, or the allele, for the different variant that a person has, by measuring the length of the genetic paragraph. Longer paragraphs means there's more repeats. But what we really care about aren't alleles. We care about the pair of alleles, which is the genotype. Genotypes are balanced in that in each cell you'll have one allele from your mother and one from your father for that same location in the genetic encyclopedia. And if there are about 15 different alleles in a population at a particular location on a chromosome, or a locus, then there'll be about 100 pairs of alleles. And that creates a huge number of possibilities just at one locus. If there's 100 possibilities, maybe 10 are very common, and you multiply it, say, 15 different locations, 100 times 100 times 100, you'll end up with like a 1 followed by 30 zeros. You have a huge number, like a trillion possible genotypes if you look at 15 different locations. Think of them as a list of pairs of numbers. At the first locus, there's a pair like 10, 12, what you've inherited from each parent. The second location on a different chromosome that's independent is another pair of numbers. So it's about 15 pairs of numbers. And there's a trillion possibilities, fewer than 10 billion people on the planet, and therefore these genetic barcodes are relatively unique. How do scientists find out what your genetic barcode is? What is your genotype? What is your pair of alleles at each locus? They begin with DNA evidence that they extract from biological materials. They amplify it using a copying mechanism called polymerase chain reaction, or PCR, that keeps doubling what's there, 2, 4, 8, 16, 32, so that after 28 cycles of copying, you have millions and millions of labeled copies that a laser beam can pick up as a genetic analyzer is separating these quantities of amplified DNA by their different sizes. More repeats, a longer allele, will be further downstream. So looking at this cartoon picture on the left, there's DNA from one person, like a cheek swab or a lot of blood. The lab extracts, amplifies, and detects the signal at this 1 out of, say, 15 locations. It's all done simultaneously in the tube for all the different locations. And what you'll get is a picture appearing as an electropherogram. That's the data where at the location where 10 alleles are supposed to be, there are 10 repeats, there's a peak. And then the allele that has 12 repeats is longer because it has an extra 4 plus 4 words repeated. And so since there's more words repeated, the paragraph becomes longer. And that longer signal is detected where it's indicated as 12 in the center. And so when a scientist sees this data, they can immediately begin to interpret it. Of the 100 possibilities that there might have been of 5'5", 5'6", 5'7", 7, 13, and so on, the data with single source DNA is compelling. Those 100 possibilities just collapse down to one possibility. When you look at the data here, there's a 10 and a 12, and a person can see it, a computer can see it, and from this, it can be inferred that the genotype is a 10-12 pair. That's the only thing that's in the data. It's the only explanation for the data. And so with single source DNA, the interpretation is very easy. That reduction of 100 possibilities down to one possibility is what data does in science. Data reduces possibilities and puts the weight where the information is here. Here, all the information is on one possibility. And based on that, a scientist can develop a statistic. A comparison can be made to the 10-12 allele pair that you see on the top right from evidence and compared with a known genotype. If a defendant or a victim's genotype is also a 10-12, then there's a match. The probability of a match is 100% or 1. And you divide that by how rare the genotype is. Say it's present in only 5% of the population. Like a random person would have a 10-12 genotype with 5% probability, out of 20 people in this room. Then dividing 1 by the rarity, which is 5%, 100 over 5 is 20, and that would be the match statistic. So with single source DNA, the NAS is completely correct, National Academy of Sciences. It's very straightforward, incredibly reliable, and there's really no issues. However, most DNA evidence is not DNA from one person. And there's this little comment on page 100 of the National Academy of Sciences report, which notes that when you have mixtures of 2 or more people, there could be issues in how you interpret the data. And that's what I'll be talking about for the remainder of the hour, is the interpretation of the usual DNA evidence that's seen in criminal cases, which is mixtures. So what is a DNA mixture? It's more than one DNA individual contributing to the mix. So you have a first person, maybe it's a victim, a second person, maybe a perpetrator, maybe a third person, like someone the victim knows. And they all mix together into this melting pot of DNA, and the task of figuring out what's in there can become more difficult. A typical example would be a handgun, where maybe 4, 5, or 6 different people have left detectable trace DNA on a handgun, and it definitely does not look as simple as 2 peaks. So suppose that there are 2 people who've contributed to a DNA mixture. Maybe we know that the first genotype is a 10-12 for other reasons. I'm simplifying to explain what's happening. The purpose of the interpretation is to separate out the genotypes. So on the left, you see a cartoon that shows there's 2 people present, not just 1. That means that the data in the center may have more than 2 peaks. In fact, here there's the the alleles, or the genotypes overlapping from two people. And now the task is to separate out from the data what are the genetic types at this locus for the two contributors. Assume we know that the first person has a 1012, that's indicated in brown on the slide. Then the question is, what's the second genotype? And if you walk through the possibilities of what alleles are seen in the data, what alleles aren't seen in the data, in this example, it must contain an 11 allele, and it can't contain anything but a 10 or 11 or 12. So in a simplified example like this, there would be three possibilities, as shown in the bottom right in blue: the 10-11, the 11-11, and the 11-12 as the genotype possibilities of the second person. This is actually a lot of information. The 100 possibilities of what you could have had have now been reduced to just three. And in science, it's okay to have multiple possibilities and some uncertainty. We just have to ascribe accurate probabilities to those. And so here are some probabilities, say 20%, 30%, 50%. They add up to 1 or 100%. And in this separation process, a second genotype is produced that contains identification information. It doesn't have to be unique in the sense of just an 11-11, but it contains a lot of information. So now that the separation's been done, as shown on the right, a comparison can be made afterwards, objectively, to a known genotype. Suppose that known genotype is an 11-12, which is the bottom entry on the list that has a 50% probability. What would happen? What does that mean? Well, the match statistics still work with DNA if you separated out the genotypes. What basically happens is that now at the top part of the equation, the chance of a match is no longer 100%. The probability of the genotype shows that it's 50%. So on the top is 50%. The bottom of the equation is the probability of a coincidence, which is still 5%. So here the match statistic, instead of being 100 over 5, would be 50 over 5. You have half the probative force. You have a match statistic of 10 instead of 20. So by separating out genotypes, you still can get match statistics that are mathematically sound, can be accurate, and explained to juries. So I'd like to walk through this with a case that we had in Allegheny County, Commonwealth versus Ralph Skundrich. And this was about 15 years ago. There was a serial rapist. He was an interesting individual in that he was both a white-collar and a blue-collar criminal. He was actually caught by his forging activity, and then he lit up a number of states on the East Coast of the U.S. Because of the rapes that he'd committed when he was doing robberies. And so in this particular crime, he threatened a college student with a gun, and the victim's jeans and T-shirt contained biological evidence. The crime lab in our county took that biological evidence, and they extracted and amplified and separated out by size the DNA. And once Ralph Skundrich was identified as a suspect in a CODIS hit, then we could start analyzing the evidence. This is what evidence looks like from 2002. At the time, there were 13 CODIS loci. This is one. It's on chromosome 13. And this is what the original data looks like. That's the starting point for all DNA analysis, whether it's done by eye or by computer. You begin on an x-axis that has size. As you go from left to right, the size increases. The vertical axis is the quantity of each allele, of how much DNA is present for each variant. And here you see a pattern. And the pattern has two tall peaks of an 8 and 11 that actually correspond to the victim. And it has two much smaller peaks on the bottom right that are a 12 and a 14 that correspond to someone else. And the task is to separate out those genotypes. So using a computer, which we were asked to do, I think this was actually the first case in Allegheny County that we were sent at Cybergenetics. This is what the computer looks like. On the left is a user interface that lets a TrueAllele operator enter data, ask questions of the data, and then the computers in the center go off and solve it. They don't know what They don't have Ralph Skundrich's genotype. They don't know what you're looking for, and they'll separate out the data into the different genotypes that are possible based on the data, give them probabilities, write the answers back down on the computer in the center on a database, and then afterwards, a user can make a comparison to 1, 10, or 100 different people to develop match statistics. This is how the computer thinks in brief. What the computer does is it proposes answers and compares them with the data. Suppose the computer proposed that there's one person present with a genotype of this 8 and 11 is 2 tall green rectangles, and then it proposed that there's a smaller amount, maybe only 10% of that, of a second person shown in the blue rectangles, and their pair of alleles is a 12, 14 in a much lower quantity. Well, then the heights of those colored proposed peaks very much explain the underlying data, and that would have a high probability. But the computer will try out another 10 or 100,000 possibilities, moving rectangles all over the place to work out the probability of anything that those genotypes could be, even if they're not in the data. It's just using the data to determine what's more probable and what's less probable by trying to explain the data and using all the data. It's just whatever's there, it's trying to explain. The result is a genotype. And here we see basically a list of, shown on the horizontal axis, of 3 possibilities, an 8-12 with 1%, an 11-12 at 1%, and a 12-14, which corresponded to those 2 very small peaks that we saw that wasn't a victim, with about 98% probability. The other 97 possibilities also exist, but they have much lower probability. And the height of the bars here show what the probability is on the list. And importantly, when the computer solved this, it did not see the suspect. It was just doing this from the data and nothing else, ab initio. So a genotype like this is produced for each of the 2 contributors that are in the data, and it's done for each of the 13 locations. So there'd be 2 times 13 or 26 genotypes. This is one of them. And now in the end, what's reported on in court is a DNA match statistic. How is a match statistic calculated? How do we answer how much more does the suspect match the evidence than a random person? What the math lets us do is now for the first time we introduce a person's genotype. Suppose it's a 12-14, which happened to be Mr. Skundrich's genotype. What we do is we compare what the evidence is saying that that contributor is, which is 98% probability, divided by the chance of a coincidence. So here that 98% divided by 3%, which is how common the 12-14 is in the population, gives us a match statistic of 35, which is inclusionary. If you moved one to the left and you looked at that 11-12 in the center, where there's a much greater chance of a coincidence than what the evidence is saying is an 11-12, maybe 10 times more, that would be exclusionary information. The number would be 1 in 10, and would contribute to saying that an individual with an 11-12 genotype is not there. So all this is done by determining what the statistic is to determine the extent of inclusion. This is done at all 13 locations. They're listed on the left, and the length of the bar is the extent of the match statistic, here on a scale of around 1 to 100. And we see that Mr. Skundrich is giving match statistics that are inclusionary at these loci. Because the genetic locations are independent, there's a product rule, and that product rule means that we can multiply or take the product of the probabilities or the lengths of these bars at each of these genetic locations to get the total number, which in this case is around 2 quadrillion. And looking at the math, we can make a very simple statement that sounds almost like ordinary single source DNA. And the reason is because we've separated out the genetic types. And to answer the question about the suspect and the evidence, the way a report would read is that a match between the genes and Mr. Skundrich is 2 quadrillion times more probable than coincidence. And all those words are the plain English meaning that are also the math meaning. that are describing the probative force numerically of that match statistic. Since the number is a quadrillion, that's inclusionary, as opposed to 1 in quadrillion, which might have been exclusionary. A similar calculation was done on the T-shirt, same method, separating out the genotypes, making comparisons to Ralph Skundrich's genotype, working out the stats at each locus, multiplying together, and the statistic for the T-shirt was 4 quadrillion. So there were 2 different items of evidence, both producing strong match statistics, and Mr. Skundrich was convicted and sentenced to 75 to 150 years. On this crime, he has a lot of other crimes that actually involved in the Butler County trial as well. And according to the prosecutor, Jan Necessary, this was a pure DNA case, and Judge David Cashman has basically said his days of torturing women are over. Why did we get this case? Why was it sent to cybergenetics in the first place? Why didn't they just use whatever the crime lab was producing as a match statistic as a result? The reason is that when crime labs interpret DNA mixtures or low-level DNA, they discard a huge amount of data and either produce no result at all or an artificially low match statistic. And this is how it happens. They apply a threshold. Threshold means at some peak height level, maybe at a height of 50 relative fluorescence units, that's the scale on the left, or 100, 150, whatever their level is, they apply a threshold. Why do they apply a threshold? Because it worked really well with single source data 15 years ago. Why would you apply it to a mixture where there's no theory behind it, it's never been validated, it seemed like a good idea at the time. And that continued as tradition. I guess tradition is probably the best answer. What happens when you apply a threshold? Well, you can discard information, but you're starting off with this pattern of 2 tall peaks, 2 short peaks. It's telling you a lot, helping your eye or a computer separate the data, but the threshold removes those distinctions. Any peak that's over the threshold is in. Any data, any evidence peak that's under the threshold is out. So either it's on the list or off the list. And you end up with 4 different alleles, the 8, the 11, the 12, and the 14. Now you don't know how much of each of them is there. And you look at all the combinations, you get 10 combinations shown on the right. And instead of being able to pick out the minor 10% contributor as that 12, 14, this method says they're all the same, all 10 of those. So by reducing the possibilities from 100 to 10 instead of to 1, it has a lot less information. How much less information? We'll quantify it right here. There's a very simple formula, which is one of the reasons the method's popular. You put in the numbers of how prevalent each allele is in the population, add them up, square it. Easy to do by hand, easy to do with computers or PopStat software provided by the FBI. And here, the match statistic calculates out to 1.7. Well, it's bigger than 1, but the true answer is 35. So you're losing information. 1 means we don't know. And bigger than 1 means more inclusionary, less than 1 is more exclusionary. And looking at what happened at all 13 loci, the blue bars, the long bars, are showing what the computer obtained as identification information, and the short red bars at each locus on top is the information obtained using thresholds and applying them to DNA mixtures. You can see that you're losing pretty much all the information at every locus, which is why when you multiply the numbers together, instead of getting 15 zeros, numbers around a quadrillion, you're getting 5 zeros, numbers around 100,000. And the result of losing all that information is that in more difficult cases, you may end up with no information at all. The number may not be 100,000, it might be 3 or 13 and have very little probative weight, and most often nothing is even reported. So the brief answer is yes. Why, how much, to what degree? That's what I'll be talking about. So the first time sophisticated computing was used on a DNA mixture that gave an accurate answer to what a match statistic was was in court in 2009 in the Commonwealth versus Foley case. And I'd like to tell you about that case. In Indiana County, dentist John Jelinek was found on his living room floor, exsanguinated from multiple cuts and wounds. He'd been slashed with a knife and with glass, and his blood was spattered all over the unsigned divorce papers of his estranged wife, who was living with State Trooper Kevin Foley. It's a very dramatic story. Books, movies, TV shows about this. I'm going to just tell you about the DNA. Kevin Foley was charged with a crime, and the main physical evidence was a match statistic of 13,000 produced by the FBI's analysis of a mixture. What was this mixture? Under the decedent's fingernails was a trace amount of DNA. 93% of it belonged to the deceased dentist, and 7% belonged to someone else. 3 different match statistics were calculated. The FBI stat using a combined probability of inclusion, which is called CPI. It's the main statistic used by crime labs for the last 15 years and still today, and it's supported in the FBI's PopStat software, gave a result of 13,000. At cybergenetics, shown on the bottom in blue with TrueAllele, the number that we reported was 189 billion. And I drew out the zeros so you could see the difference in orders of magnitude of powers of 10. And another consultant, for good measure for the prosecution, came up with a number in between that was 22 million. And that led to questions. Why are there different match results, and why do they differ, and what's reliable? In overview, this is what the different methods were doing. The first method, CPI, or combined probability of inclusion, did not use the fact that the victim's genotype was known, and it applied a threshold to the quantitative data and just used in or out. The second method from Boston University assumed the victim was known, but again, didn't use the quantitative data, it used thresholds, whereas TrueAllele, the computer program, did use both sources of information, the victim's genotype and the quantitative data. So that's one difference between the methods, is the data that they're examining. A lot of different techniques go into the analysis, but that's the difference in the data. There was a Frye hearing with President Judge Martin presiding, and these are some of the factors that were discussed. I won't mention them all, but these were documents that were provided of different papers from the scientific literature, maybe 60 papers, that talked about genotypes, quantitative STR data, publications, and why the notion of using all the data with statistical computing was generally accepted in the relevant community, which the court accepted to be those scientists that develop and test these methods, not the practitioners. These are some of the pictures that I discussed with the judge for a few hours during that Frye hearing in 2009, February, I guess exactly 7 years ago. The first was, what is a threshold? What do we mean by a threshold? And so this was a picture that I showed, and it's a picture of a young man, and you can't tell too much about him because it's all monochromatic. It's either black or white. That's what a threshold does to information. But when we showed the grayscale picture, it was clearly Jimmy Stewart. The reason it was clearly Jimmy Stewart is because, as shown on the bottom left, Jimmy Stewart's statue is outside the courthouse. So the judge would see this on a daily basis. And the Jimmy Stewart Museum is right next to the courthouse by the parking lot. And this is actually taken from It's a Wonderful Life. So when you get rid of quantitative data, you get rid of a lot of information. And we see this visually, in this case with a picture, more than by looking at arcane peak heights. This is a scatter plot that shows points moving up to the right. What it shows is that for each point, the horizontal value is the amount of DNA, the vertical value is the match statistic. We see a straight line that connects and relates them. Why was that important? A main defense point was that all match statistics should be the same, and we all expect them to be 13,000. But we were finishing up a paper at the time based on totally independent data of known composition, which showed that there was this relationship when we used TrueAllele. And in fact, by looking at those dotted lines, we predicted with 12 genetic tests, 12 loci that were used, that we actually expected a match statistic in the hundreds of billions or around a trillion based on science, not based on what some other method might have done. That was ultimately published later that year. So there was an expected scientific result that based on validation studies, not just based on the evidence in the case. Judge Martin admitted TrueAllele into evidence, and ultimately the case went to trial. I was able to give expert testimony. We explained how the computer worked, what it was doing. The prosecutor was at the Attorney General's office, Anthony Krastik. It was one of his last cases. And I had come in with maybe 40 printouts that I wanted to show, and Tony judiciously chose 4 of them and threw out the other ones. And so this was a good move. And so this is what we talked about. And a lot of it had to do with addressing the question of shouldn't all methods give the same answer? And no, if you use more of the data, and a computer can use more of it and do more processing, calculate more variables with it, and explain the data better, then you don't expect to get the same answer. And I gave an analogy of a microscope slide. I mean, you can look at the same microscope slide, but whether you're using a magnifying glass or a microscope, your measuring instrument on that data will give you different information. So the jury convicted the trooper of killing John Jelinek. And the main evidence was the evidence provided by the dentist having reached out and scratched the forehead of the trooper and gotten a little bit of trace evidence under his fingernail. The trial decision was appealed as Mr. Foley was serving his life sentence. And in an appellate court ruling that was published in the beginning of 2012, the court found the TrueAllele method was reliable and established a statewide precedent for TrueAllele in the state as a way of reliably interpreting DNA evidence. So I'd like to talk with you a little bit about the peer review process. And the reason is that peer review is often mentioned in court, and it's helpful to know exactly what it is. The concept of peer review is shown at the top of the picture. Scientists, a team, do research. And then after they've done all their research, they write a manuscript, scientific paper, and they describe what they did and what the results are. Usually it's methods, materials, and results. There may be other sections where they discuss the findings as well. Then as shown in the center of the slide, the manuscript is sent to the journal editor of some scientific journal, and then an independent and anonymous review by at least 2 scientific peers is done, shown on the right, and then the result of this is either accept, reject, or edit. And usually it's edit. Reviewers would like some changes. The editor communicates back to the authors. This can iterate a few times. It might happen once. It might happen a half dozen times. And if ultimately, when everybody's satisfied that the work is worthy of being published in the journal publishes the peer review paper. This is different than writing a report or publishing it on the internet or just having a report and submitting it in court. This is what the peer review process is: independent, anonymous review by other scientists. This is what a peer-reviewed study looks like to establish the reliability. This is a recent validation study on TrueAllele. This paper is 10 or 15 pages long. It's printed by the journal. It's gone through the peer review. This one has a title like TrueAllele genotype identification in DNA mixtures containing up to 5 unknown contributors. So when we test these systems, we test them under more and more harsh conditions. It's not just 2 contributors. This paper's on 5. There's usually an abstract that describes the main findings that may be read out in court by someone one day. There are the authors of the journal. Here is the Journal of Forensic Sciences, which is the main American journal of forensic science. That's what a peer-reviewed paper looks like. Papers may be open access, like this one is, so anybody can freely download them and look at them. There have been over 30 validation studies on TrueAllele that look at different aspects of how it analyzes data and gets results in mixtures. They've been done by crime labs, they've been done by cybergenetics, they've been done through collaborations. 7 of them have been published, and there's an interesting distinction between 2 types of studies. The top 4 studies that were published were done on data of known composition. That is, scientists in a laboratory synthesized the data from DNA that they knew what the genotypes were, mixed them together in different proportions and in different amounts, and then that was the data that was presented by to the computer to be separated. That has the advantage that there's a suggestion that we might know the answer. Obviously, there can be mistakes in how the lab prepares it, but we have a better idea that we're going to, we know what's present in those samples. The bottom 3 are done on actual casework samples. And that's also important because usually the quality of the data that's scraped out of a body cavity or off the floor is much, much worse than what you make in the laboratory in a test tube. And so it's useful to have both types of studies. In the case of a computer program like TrueAllele, we have lots of validation studies. In the case of methods like CPI and threshold methods, there's never been a validation study in the last 20 years for them. These are the types of study axes that are looked at. Each paper will typically look at 5 or 6 different axes. Some will be unique to that paper, looking at the effect of different parameters. Comparisons are often made with human results on the same data to give a sense of how people are doing. These 3 axes, though, appear in most modern validation studies. The first is called sensitivity, and that means we're looking at if someone has truly contributed their DNA to that mixture, are we finding it? And to what extent is the match statistic? The second one down is called specificity, and that's if an individual has not contributed their DNA to a sample, are we properly excluding them? And what's the false positive rate when they're not excluded? The final axis is reproducibility. And this appears because with complex statistical software that's like TrueAllele, and you can be running 100 dimensions to solve the problem inside the computer, then statistical algorithms have to be used that use sampling. Think of it like a voting algorithm in that every time you sample, the vote could be a little bit different. It should be roughly the same, but you don't expect it to be identical. So the degree of that variation is measured as well. So those are the usual axes that you would see in an admissibility study. And the way these studies are used now enters into the notion of rules of evidence. And I'm going to talk about reliability briefly. So there's a PA Rule 702 about who can qualify as an expert witness. Usually with DNA, the expert satisfies criterion A, that they have some scientific or technical knowledge that is beyond what probably anybody in the court building might have. That's why they're wearing a white coat, talking about what they're talking about. And also criterion B, that it's relevant to the case. That's actually some DNA evidence that's interesting. But if there's some sort of an admissibility hearing based on reliability, the question will come up of what's the methodology, and is the methodology they use generally accepted in the relevant field. In federal court in our state and in most of the states, there's the Daubert decision that determines the criteria for determining reliability, and it establishes the judge as a gatekeeper. And what's interesting is that the first 4 criteria are largely satisfied by extensive validation studies. If you're testing software on real data, then it's been tested, it's been peer-reviewed and published if you've gone that route. The error rates have been determined if you're validating it the right way, and the standards and controls of how you run the software come out of the validations, like running everything in duplicate to make sure you get same answers. What Daubert shares with what we have in Pennsylvania is the general acceptance in the relevant community, and that's the Frye standard from 1923 from the District of Columbia Federal Court. And here the standard, in a very short decision about a lie detector test that was ruled inadmissible at the time, is determining, as it says towards the bottom, is that has the result of the deduction been based on methods that are sufficiently established to have gained general acceptance in the particular field. So that's the Frye standard that we have in Pennsylvania that with TrueAllele was determined in the Foley decision. What does it mean that there's the scientific community? Well, there's peer-reviewed papers that helps determining the scientific community. In the case of TrueAllele, it was assessed throughout the country. So there was an independent assessment of the results presented by the New York State Lab in Albany over 1 or 2 years. And that was accepted by an independent committee that looked over about 600 megabytes worth of data and reports, summaries, and so on. So acceptance by a forensic science commission after review is one way of having a general acceptance. Another is its use outside of forensics. TrueAllele was used 10 years ago to reanalyze the World Trade Center disaster data, working to reassociate through DNA the 18,000 victim remains with the 2,700 missing people through their genotypes in this objective process. You infer genotypes from the victim remains, infer genotypes from kinship information or personal effects, and then compare them to determine the extent of match. So at this point, TrueAllele, we started developing it in 1999, and we went through 10 years of 25 versions of expanding the statistical model and testing it until we couldn't see that it was doing anything stupid or wrong through testing. And so after that development phase, it started being used in court. It's been admitted after Frye and Daubert challenges in 7 states and some foreign countries. There are crime labs using it regularly across the country. It's been used in over 500 criminal cases in the US for both prosecution and defense. And there have been over 70 criminal cases in Pennsylvania across 17 counties, which I list at the bottom. Your county might be up there, I don't know. It's widely used, even though it's challenged, and then we use science and studies and general acceptance to overcome those challenges. So what happens when you have a scientific method that's generally accepted as reliable and published as reliable, particularly in Pennsylvania when you're not going to be getting a Frye hearing? You have to go to something else, because the 702 isn't going to help a defense attorney in that. Usually it's a defense attorney. So this is what source code is. Let me tell you a bit about source code. When computer programs are written, they're written in a programming language that people can read, that programmers can read, share, understand, that provide a set of instructions in a human readable form that describe what the program's supposed to do. And then as shown in this red arrow, a compiler program translates that human readable development, like the blueprints of the software, into an executable program that you actually run on a computer or like on an iPhone. Those are executable programs that are running. And that's what we test. When you're running your iPhone or a computer program or your car, which has a half dozen computer systems on it, you're running the executable code shown on the bottom in blue. And for those programs, input comes in, as shown on the left, output comes out of those programs. That's the software that's actually running in the real world that we test. When testing is done in validation studies, nobody has source code. Even the developers don't use it, because the question is, does the executable program work the way it's supposed to? Now, in a criminal case, a discovery request can be made under the Rule of Criminal Procedure 573, unless the software was developed by a government and the attorney or the state is in possession of the source code. We move from mandatory disclosure to section B2, which is discretionary disclosure. And there are three requirements that have to be met. The first is a showing that the material, what's being requested in discovery, is material to the defense preparation, that the request is reasonable. It won't put companies out of business or you can't ask for somebody's beating heart. I mean, you could, but most judges in the US wouldn't give it to you. And it has to be in the interests of justice. There has to be a reason why you would do this. So discovery requests can be made for something like TrueAllele source code. And the argument is usually from the defense, and we see this around the country, that the Sixth Amendment says that you have the right to confront your accuser. The source code is the accuser, and therefore we need the source code. The purpose of it, of course, from our perspective, is that it's a ruse. There's nothing you could do with it. In the case of TrueAllele, it's 170,000 lines of dense programming language. It takes the experienced programmer about an hour to read 10 lines of software. So there's nothing you could really do with it. Moreover, we always offer to let the other side, whoever they are, run the program for free and test it on the internet. So you can always test it. And then the argument that gets strange is that in science, testing isn't what you determine. You read source code, which actually doesn't make too much sense to scientists. The problem is that with something like TrueAllele, the source code is a trade secret because if we disclosed it, cybernetics would go out of business because of competition. And trade secret is defined on the slide. And this was actually handled in Pennsylvania in the Foley appellate ruling from 2012, in which they wrote that scientists can validate the reliability of a computerized process even if the source code that underlies it isn't available to the public. And with proprietary software like TrueAllele, it wouldn't be possible to market it if it was given away for free, and it's been tested and validated in peer-reviewed studies. So the appellate court agreed with the scientific community in that testing is what determines reliability, not reading programming language text for 10 years. And this comes up, and one court granted it in California. It was quickly overruled by the appellate court. The judge in that case said that he knew in his gut that the Sixth Amendment had to apply, but the appellate court said the law didn't agree with that. And so these discovery requests are denied, and I list 6 states where this has happened in the US. Moving on to Rule 403, I'd like to talk about excluding DNA evidence based on relevance for prejudice, confusion, waste of time, and so on. So under the federal and state rules of evidence, in the state rule, significantly is not a word that's in the phrasing, a court may exclude relevant evidence if its probative value is outweighed by a danger of one or more of the following, and it includes unfair prejudice, confusing issues, and so on. So there's a balancing test between probative evidence. DNA is presumably probative. It's supposed to prove something one way or the other, versus the danger of unfair prejudice, which DNA definitely has, because if you start whispering DNA, it's pretty clear the guy must be guilty, and that's the end of it, if you don't know more. So it's an interesting test, and I was actually involved in a case in Butler County with Judge McCune, who ruled DNA inadmissible in a very interesting circumstance based on Rule 403. The way it went like this is that a man was accused of sexually attacking a woman in a house, and the evidence that was brought forward was 2 types of DNA. The main type of DNA was called YSTR. This is DNA that's passed along paternal lineage. So it's whatever you've inherited from your father or your grandfather. What made that interesting is that there were only 5 men in the house that night, and all of them shared the same grandfather. So they all had the same YSTR profile, and the DNA couldn't have distinguished between any of them, even if a crime actually had occurred. Moreover, the house was saturated with their DNA, so the DNA may have just arisen because DNA got into clothing in a person's house. So in that case, since the DNA couldn't distinguish between anybody, it wasn't proving anything. And that was the main DNA, and I reproduced part of his ruling on the right, and the DNA was excluded as misleading, confusing, and prejudicial. So this case went up, I think, on an interlocutory appeal to the Superior Court, who agreed with the trial judge. And I think Mr. Booher, in the end, was not convicted. So if the DNA is not relevant under Rule 403, it may not be, it's the possibility that it's not admitted. So I want to tell you a little bit more about crime lab DNA interpretation, how it's done in crime labs in the US for the last 15 years and today. So there's this concept that you get this data, an expert in a white coat from the government, and a match statistic. It's 1 million, whatever the number is, and that this is completely objective, and all you're looking at is that match statistic that gives the probative force of the evidence. But what happens on the way to the match statistic? The first step is that the data is adjusted. Thresholds are applied. That's pretty universal in crime labs. Exceptions are made. Artifacts are looked at and decided, are these extra peaks expected to be there from what we know about the amplification process? Or maybe there's peaks that are missing that could have been there because of a phenomenon like dropout. So the first step is to take the quantitative data that comes off the genetic analyzer and play with it. That's what happens. A human analyst determines what will go into the program. The second step, is written in most crime lab protocols, is to then determine, is the defendant or another person included in the data that remains? So they look at the list of allele pairs from the defendant, they compare it with a list of alleles from the adjusted data, and they decide, as an expert decision, is the person included? And only after that decision's been made do they then calculate a statistic. And then that's what you see in the report, but you usually don't see the first two steps because that's somewhere in a protocol. That's not what's reported. And also there are many labs whose protocol is, if the computer doesn't agree with you, play with it some more. Because you're the expert, and you must have been right, and the computer is what's not right. So the problem with this is that it's not an objective science. The DNA isn't like with single source DNA, or using TrueAllele. It's a human examination process that the NAS report decried in 2009 for the other fields of forensic science. The statistics and the reporting are just the end result of a number of qualitative subjective manipulations, and it's unclear if this has any scientific basis. There's never been, to my knowledge, any validation study, published or otherwise, by proponents of methods like combined probability of inclusion, CPI, and the PopStat software. It's just a given, and you can kind of see how that works out in court. On cross-examination, how far can you get? Well, why are you using this method? Well, it's the standard accepted software in a lab. We're following the protocols. Great. And have you validated your method? No, but we're using accepted software. And where did that come from? Comes from the FBI. And I've watched this, and usually the whole discussion stops. I mean, if you took it further and said, has the FBI validated it? The answer would be, I don't know. It would have the same effect. So most of the mixture interpretation is being done using subjective processes with unvalidated methods. And this has been coming to light in the last year around the country. In Washington, D.C., the crime lab was shut down over DNA mixture interpretations and the CPI method, this inclusion method. The top 6 brass were fired, and now everything's running again with new people installed who are former FBI employees. In Texas, they realized there was a problem when they reanalyzed all of their mixtures because of population database change. The database change of what random would be didn't affect things very much, but there had been an intervening change in the methods the crime labs used that introduced a second threshold that came in 2010, and so the numbers were very different. The average number had been 1 million, it reduced to 100, and that question was, well, if the real match statistic was 100, did that mean we'd overstated match statistics before? Were there potential false convictions? We'll look at that in a second. So there are questions that come up about methods that have been getting out of the lab and into the public. For how long has the forensic science community been on notice that there could be a problem? Well, the first study conducted by the federal government and the Department of Commerce, the National Institute of Standards and Technology, which used to be the Bureau of Standards, did a study on 80 or 90 labs. What they did is they had the same simple two-person mixture, and laboratories who participated looked at the identical data, and this is a slide from a talk given 11 years ago by Dr. John Butler at NIST. And what they showed is that on the same data, there was vastly different results. A large number of the labs reported the data to be inconclusive, and so they wouldn't give any number at all. And on the other, roughly half of the labs, the match statistics were all over the map. The numbers ranged from tens of thousands, which is a 1 followed by around 4 zeros, up to hundreds of trillions, which is a 1 followed by 14 zeros. So there's 10 powers of 10 difference between tens of thousands and hundreds of trillions, or nothing at all if it's inconclusive. What happened after this is the FBI revisited its standards and said there's a lot of variation in data, which is true. But instead of modeling it with computers, they decided the best thing to do would be to introduce a second threshold, usually about 3 times taller than the first threshold, and just keep throwing out more data. And what ended up happening is that if you had 15 loci and you threw out what ended up being around 2/3 of the data, now you'd only be reporting on maybe 5 loci. So your match statistic would shrink by 2/3 from 6 zeros or 1 million down to 2 zeros or 100. And so a lot of DNA evidence wasn't used. With a lot of, and we have papers on this that I think I've included or on our website. Other people have written about this. But it didn't become more reliable. The whole issue was false inclusion. So after these methods came out, NIST looked at it again in the 2013 in their Mixed 13 study, expecting all problems to disappear. And that isn't what happened. So here's a 3-person mixture reported by Dr. Michael Coble from NIST. This is 100 labs who've looked at a 3-person mixture. 70 of them, shown on the bottom, falsely included and gave match statistics ranging from 10 to 350,000. There were other labs, maybe a quarter of them, that said it was inconclusive, and 6 labs properly excluded the suspect based on their protocols. The larger problem is that these numbers don't even have any probative value. This is a paper that I published. There's been a number of papers by researchers about problems with CPI and the match statistics used in hundreds of thousands of DNA mixture evidence items over the last 15 years. But what this paper showed, the title is Inclusion Probability for DNA Mixtures is a Subjective, One-Sided Match Statistic Unrelated to Identification Information. There's actually no statistical, well, very little statistical relationship. And is essentially a random number of inclusionary values that didn't add much meaningful information. So what that means is that an analyst goes through a subjective process of choosing the data they're going to use and discarding much of it, then deciding if a defendant is included or not, and then to confirm what they've decided, they then put it into a program. That would be great if the program actually correlated with identification information. But if it's just a random number from an unvalidated program, then what's it doing in a court of law? And so this brings up the question of the relevance of the CPI match statistic. It's unvalidated. It's not related to identification information. It may have very little probative value if it's just a number that's always inclusionary, that's not related to identification, that will always agree with whatever the analyst wants or what their conclusions are, because they've already reached that conclusion and it can't do anything but give an inclusionary number. But There's a lot of potential unfair prejudice calling that DNA evidence. It's not what a jury or a court thinks of as DNA, that it's a random number resulting as the outcome of some subjective process. The larger problem than even that potential injustice is inconclusive mixtures. Most mixtures are called inconclusive by crime labs. That means they can't interpret it. It also often means that somebody sends it to cybergenetics, or a crime lab will purchase technology that can do more. What does an inconclusive mixture mean? It means that the data are sitting there as quantitative data, but the lab's protocols won't let them analyze it. Maybe the signals are too low. Maybe everything's under threshold. Maybe the data is too degraded. Maybe there's more than 3 people or 2 people in the mixture. Depends on the laboratory. So for example, handguns often won't get analyzed by a lab at all. They'll generate the data, but once they see there's 4 people in there, there's nothing to report. So this is an example of such a mixture from a case that we had in Allegheny County. And like most labs, the crime lab can't do much with a 4-person mixture. Just drawing a threshold line doesn't give a correct answer most of the time. And so based on the fact that there was too many people who had touched the handgun and left their DNA, no conclusions could be drawn. So now we have 2 defendants, and they're going to trial, or they're not going to trial, whatever the district attorney wants to do with this case, but they have no DNA on the handgun. So we get this. There's a computer reanalysis that's done, and in cybergenetics TrueAllele report, the match statistics are informative. What happens? Well, starting on the left, TrueAllele unmixes the mixture. So there's a complex mixture with a lot of peaks and data. It separates it out into the genotypes. They're fuzzy genotypes. They have probability, but they're informative. Fuzzy genotypes of the 4 different contributors. And then that's done objectively. The computer is not looking at any of the suspects. After that objective computer statistical separation is done into the 4 genotypes, then a comparison is made against the various suspects in the case. Now what we find is person A is excluded. And what the DA does is it drops charges. So that's what happens when you have real information. Same data, but better interpretation. Then there's another individual, and he was included with a match statistic of around 500,000. And he ended up accepting a guilty plea, and he went to jail. So if you're not interpreting DNA evidence and you're throwing out most of it as inconclusive, it's not just that there's court costs, waste of time, and so on, but the information that can acquit or convict is just not available to society. In Allegheny County, I just heard about another case this morning. Of the 40-plus cases that we've done, I've testified 8 times. 3 of the DNA results that I've heard of have resulted in exonerations. And these are listed as now 20 guilty outcomes. What's interesting about it is 12 of them are guilty pleas over a wide range of evidence, often guns and clothing, for a wide variety of crimes, usually pretty violent crimes and serious, involving rape and homicide, child rape, and so on. The sentences range from a year to life, whatever they happen to be. But the point is by having DNA identification information in the courts that's accurate, there's a lot of unnecessary legal work that can be avoided because the information's there. You know who's contributed their DNA and who hasn't. I'd like to make a quick note about post-conviction relief. There's a Title 42, Chapter 95 has a PCRA provision, and the usual kinds of filings will have to do with ineffective assistance of counsel. But what's been interesting, and I've been involved in these in other states, is if potential exculpatory evidence was not available at the time and it became available and it would have changed the outcome, then a hearing can be granted. And as I show in the middle, TrueAllele analysis of DNA evidence that had been called inconclusive but is now informative has been the basis of getting a hearing in some states, because of inaccurate or inconclusive statistics. And there was a very interesting case, Lee versus Monroe County, that I heard about from the PA Innocence Project, where involving fire evidence, it turned out that the scientific basis of the fire evidence didn't exist. A major expert in arson analysis wrote a report that said the fracture pattern was caused by water, not by an accelerant. And the defendant went through the three state levels, failed for a fourth time at the first federal level, and then at the circuit court actually was granted habeas corpus relief, and I think he got out of jail last year because the forensic evidence was so flawed that the conviction was essentially overturned. If you're interested in reading more about TrueAllele, we have lots of CLEs, articles, research papers, presentations, and so on our website. Feel free to email me if you have more specific questions